All right, we're going to look at how the cell is going to create proteins. And I want to remind you that proteins are one of your four macromolecules that is of amino acids. So amino acids are the monomer of proteins, and that's going to be important when we look at how we're going to assemble our protein and what order we're going to place these amino acids to do it. Just kind of a review that proteins also do a lot of cool biological things. They help make up structures, they work and act as enzymes, and we know those are significantly important in the cell in your body. So when we look at this, we can note that DNA, which is a blueprint of life, it has the code, the genetic code for making proteins, and it's safely locked up inside the nucleus. It doesn't leave. The molecules are huge, they can't fit through the nucleus, and we want to keep them safe because that's our DNA. It's one of the most important molecules we have. It's telling us how to make those proteins. Out in the cytoplasm of the cell, we have the ribosome. And that's what's going to use this information to make that protein. So the big thing is we've got to take that information, those directions, and move them here so that the cell can use the ribosome to actually make the protein. So what we're going to use is this macromolecule, RNA. It's a type of nucleic acid. Remember, Na stands for nucleic acid. D stands for deoxyribose because DNA is named after the sugar. RNA is no different. It's ribonucleic acid. It's named after the sugar. Both of these are nucleic acids. So each of them have that sweet sugar center. They each have a phosphate, and then they have those nitrogen bases. And then depending on the structure and what our bases are will determine this is a significant difference between DNA and RNA in terms of being different types of nucleic acids. So when I take my DNA and I convert it, or I rewrite it, or I put it into RNA, that's called transcription. And so what I'm literally doing is I'm just taking one type of nucleic acid and creating another type of nucleic acid. It's no different than if I'm reading a textbook, reading a cookbook, finding the recipe I want, and just writing down that single recipe on a sheet of paper that I can carry with me to the kitchen and make whatever I need to make. So it's called transcription because of that. I'm just rewriting it from one form to the other. This is going to happen inside the nucleus because that's where my DNA is. And the specific form of RNA that I'm making inside the nucleus is called mRNA, with that M standing as a messenger. It's messenger RNA because now I have the message that's located in the DNA, that recipe, those instructions, and it is now small enough that it can fit through the nucleus and go to the ribosome. Because you want to think about RNA, again, being that single little piece, that one portion of one side of DNA that we're writing down and taking with us to the ribosome. So once we have that made, it's going to be able to go to the cytoplasm. So some of the differences then is, well, how do I know how to make that messenger RNA? And we're going to do it according to base pair rules. Some of them follow the rules of DNA replication. So we're familiar with that G and C and G go together. We know that T and A is what we should see with DNA replication. However, we're rewriting it. So we're moving from the DNA into the RNA. So this is like one direction. And if I have a T in my DNA, I'm still going to base pair it with an A. However, if I have an A along my DNA, I'm going to create that messenger RNA using a U. So A will get noted down with the U, T goes to A, C to G, G to C. So I'm making that messenger RNA using the DNA as a template, but I'm base pairing it now slightly different. So to practice that, you have this top of your page where here I have DNA. I know it's DNA because it has T's. And then I'm going to write down, according to my new rule, to form that RNA, I'm going to use the bases that I should to create that messenger RNA. So C still goes to G, but A's go to U's. T's still go to A's, but anytime I have an A, it should go to U. And I'm just going to practice writing that down because now this is my messenger RNA. It's short, it's just a length of one gene, and it's just a single strand. It's that small little portion now that can physically leave and go through the nucleus that I can now functionally use at the ribosome to make that protein. Now, creating the protein is called translation. So transcription is I'm taking DNA to RNA, nucleic acid to nucleic acid. Translation is now I'm taking that messenger RNA and I'm making a protein. And proteins are a different type of macromolecule versus a nucleic acid. So since I'm going from one type of macromolecule to another macromolecule, that's translation. It's not like if I'm taking an English cookbook and I'm writing down in English the recipe I want, that's transcription. But if I take that recipe that I wrote down in English and I turn it into Spanish, that's translation. I'm putting it into something different. And that's going to happen on the ribosome where I'm going to create my protein. And so the sequence of my messenger RNA is going to tell me what amino acid I need and what order is it going to go in. So each of my letters means something, and I read them in groups of 3. So GUC is going to tell me one specific amino acid. AGA is telling me another specific acid. AUC is telling me yet again another different amino acid. And each 3 letters, each 3 bases is called a codon. And each codon then is coding for a specific amino acid. And this is the order it's saying I need to put them in. And when I string my amino acids together, I've got that protein. And that's how it works. It goes from DNA to RNA, and then I'm going to get that protein. We're going to get a lot of practice actually doing translation and looking at that expression, because once I have that protein, that's genetic expression. I've expressed a gene that's written in the DNA, and now I have that functional protein. Because the translation, the creation of the protein, relies on the messenger RNA, I have to have transcription first. So transcription has to happen first, then translation can happen. Now the cool thing is, if I make any changes, or if a change happens to any one of my bases in DNA, that is a mutation. Because what's going to happen is the corresponding base in the messenger RNA is going to be different. And therefore, that codon is now calling for in terms of amino acid is going to be different. And it changes the whole protein. And mutations can be good, bad, or indifferent. Some mutations are great. Hey, you get to still have drink milk. Some mutations are bad, where you could end up with cystic fibrosis. Some mutations are indifferent, and it's just a matter of like, oh, great, you've got blue eyes now. Does it hurt or help you? It's just a different way of having your eye color show up. So if we were to compare then and look side by side between our 2 types of nucleic acids, we can see that DNA, remember, is going to be the bigger, longer, more complex molecule because it's double-stranded, it's helical, and it's got all the genes located on it. And because it's so big, it stays in the nucleus. Our RNA, remember, is just a piece of the cookbook, the recipe. It's just a single gene, so it's going to be single-stranded, it's going to be super small, so it can fit out through the nucleus, and it's going to can be made out of ribose, because they're both named after the sugars that make them up. There's only one type of DNA that we use from the nucleus, but there's actually 3 types of RNA. And messenger RNA, mRNA, is the RNA that gets made in the nucleus. rRNA is the RNA that actually makes up the ribosome. And then tRNA stands for transfer RNA, and that's the type of RNA that carries the amino acid to the ribosome and fits them in place. Both types of nucleic acids, RNA and DNA, have the same base, adenine, cytosine, and guanine, but DNA is going to use thymine, where ribose is going to use uracil. And so that's a look at protein synthesis.